So we are now moving to the third work stream, clinical trials. So the team we have assembled here today, we're really happy. Amy McKee is here from the FDA. And as I mentioned, she's working with Rick. They do incredible, incredible work. So we're trying to work side by side with them as we move toward these platform trials. And then we have Laura, who's here from CRI, and that is very much because they're doing a ton of the work in immunotherapy and doing some really nice work in innovative trial design. And then Brian Alexander, who is spearheading the major glioblastoma effort, and will tell you an amazing story about how challenging it can be when you don't have a trusted third-party disease foundation that can work with you, and how he had to build his own nonprofit to start to do that. And then Clifton, who just recently ran a clinical trials panel at Brainstorm Health and did such an amazing job that I recruited him, of course. And Bill, I'm hoping that your question maybe will sneak it in at the end of this, because I don't want to leave you stranded. We'll make it work one way. Okay, all right, thanks.
So that last conversation between Allison, Colin, and Lee and Paul was the perfect transition for this conversation. Alice, you mentioned that the randomized controlled trial has been the gold standard. I just want to remind everyone that the U.S. went off the gold standard in 1971. So that does give us some context for this. And I think there's some nice parallels in there. We've come to appreciate the power of the clinical trial in terms of understanding efficacy and safety of drugs that we test. And yet, so much has happened in the last 30, 40 years where the science has moved forward very, very quickly. Our understanding of the cancer process and disease process in general has moved so quickly, changing the way we think about things. The sheer number of patients that we're dealing with, the sheer number of drug makers and device makers, and the difficulty of recruiting and accruing patients to those. So, it's not that it's either or, it's that life has changed. And so as part of that, we have all these new trial ideas and new formats, and we're going to talk about the master protocol or the platform trial or some of the variations on that theme today, which is essentially taking a good idea and making it better. My first question is for you, Brian. We have the I-SPY 1 and 2. We've got the INSIGHT trial. We have GBM Agile. We have 1,000 more. Are we in danger of running out of acronyms? Okay, that's not really my business.
No, that's
true. Okay.
It probably took longer to get some of the acronyms than it did to do the design.
So you started, let's just get right into GBM Agile and why you started this. And tell us a little bit about what it is, actually, so we understand.
Sure. So both INSIGHT's a GBM trial that's run out of Dana-Farber, which is a kind of more of an investigator-initiated study, and GBM Agile is a larger scope study that's intended to be a registration study to try to attract more drugs into the GBM space. So we did it for a number of reasons. One, I was seeing patients with incurable brain tumors and there was no trial options available. And a lot of the reason there wasn't trial options available is disinterest from the pharmaceutical community and biotech community in GBM, just bureaucracy. There might be a trial that's going to open, but it takes, a group of 50 people to coordinate their schedules over the course of a year and a half to kind of reinvent the wheel of the trial that we did last year. So there's a lot of inefficiencies in the space. So one of the things we said was we wanted to kind of, remove some of those inefficiencies so that if you're really just asking the same question or the same testing structure on a new drug, just, approve the new drug through IRB and all the kind of things rather than the whole trial again. GBM Agile also, I think by making it more of a registration trial and trying to kind of create an organization that's sustainable. The real focus for the organization is to say now we want to have like some entity in the pharmaceutical space that cares about GBM. No one else cares about GBM. I mean, they might care about GBM if like the interests of the molecule overlap with the GBM pathway. That might be a reason they care about GBM, but not inherently. So, in the steady state, we would like to kind of enroll as many patients as we can in the trial. The other thing about GBM Agile is there's two major indications in GBM. It's newly diagnosed and recurrent disease, and we use both. We kind of include both those kinds of patients in the trial and use a statistical model to kind of pool the power. So what this does for patients is that you can kind of come into the clinic and automatically be enrolled on the trial. So I think what we heard before in the last panel is the same questions as we're trying to address now, is that there's clinical research and there's clinical practice, and those have traditionally been very separate. And in clinical practice, you don't really systematically learn anything, and clinical research is very kind of siloed, answering one hypothesis. And what we're trying to do, I think, from that side is what we heard the last panel, make clinical practice more able to generate evidence. But from the clinical research side, we want to make that more seamless so that patients can kind of enroll and we can learn from as many patients as we can and give them chances to be on experimental therapies.
But just so the platform trial, though, the idea is that you are essentially randomizing in the beginning, right?
Yep.
Patients to different therapies, testing multiple therapies. And as time goes on, you learn from that information about who's responding and who isn't. Some of those therapies graduate, and some of those are dropped.
Yeah.
And the ones that don't seem to work. And so this is a way to accelerate the process.
Yeah, so I'm a firm believer in randomization for a lot of the questions we wanted to ask. Because I mean, I think if you look at the trials that have done in GBM, there have been 10 or 15 over the last 10 years, very large randomized phase 3 trials based on single-arm phase 2 data that was overly optimistic and said, like, there's this big signal, and then there wasn't when you actually did the randomized trial. So randomization is very important to me for the kind of signals that we're looking for. The platform trial is a way to kind of do that efficiently and have one control arm that's a control arm for several experimental arms rather than doing one-to-one randomization. And so still getting some of those randomization elements so we can make different inferences, but doing it in an efficient way. At the same time, we're doing adaptive randomization so that if one arm looks like it's better than other arms for a particular biomarker set, it will start weighting the randomization. Still randomized, but it will start weighting that randomization. And that randomization probability can drop to 0 so that if you don't have a biomarker that you need to have to respond to a drug, those patients won't be enrolled on that arm anymore.
So, Laura, you work with an organization that really focuses on immune therapies and has since its very beginning, and you now have a platform trial that you're ramping up in this area. It's a different set of circumstances when you're dealing with complicated immune therapies, many of which are involve new technologies like in CAR T that don't really conform to our standard small molecule approach. How are you doing this? What's your baseline for understanding this new trial?
Well, for the Cancer Research Institute, we followed the same traditional model of single trial, single question for a long time, I think.
Right.
I think with the landscape analysis that sort of quantified the busyness of the immunotherapy space with 2,000 agents in development and 3,000 trials.
Massive, 2,000 agents
it's just
in development.
huge.
It's incredible.
This is a very conscious shift then to a platform approach and master protocols. So we are ramping up a study under that is looking at it's a biomarker-driven trial that will enroll patients based on different biomarkers. I think the approach that we've taken, because the Cancer Research Institute doesn't conduct trials on its own, it funds clinical trials through philanthropic funds and through co-funding mechanisms with biopharma partners, and through access to a menu of agents. It's we've partnered with a group that has a very strong operations component and core that really have a long history of efficient trial design and execution. There are still challenges, though. That doesn't remove the difficulties in getting a trial going.
So the challenges. So one of the questions that you have people that support this old gold standard idea. The RCT is still deeply embedded in the culture, even as the FDA is changing its focus. There's still a lot of people who are whispering about this. One of the things they say is, well, there are biases that work themselves into the platform approaches. One of them is that people will wait to join trials based on the fact that as certain therapies get better and start to graduate, or they're more that the losers drop out, that they have a better chance of getting one of those. How do you deal with that?
Yeah, so I think this is an important point, is that you're trying to have a solution to a problem, not start with the solution, find the problem. So like for GBM, there may be one set of solutions that is the problem, and I think that what we've come up with is particular for GBM. that drift in time, you can do things statistically, like control for it by looking at the different ways that the control arm has been fluctuating with experimental arms over time. GBM, we don't really have this issue because patients get diagnosed and they don't have a long life expectancy and they get on a trial right away. So there's
just
no waiting.
Tell people what the life expectancy is. I mean, I'm just to put this sort of safety and efficacy framework in action. How many GBM patients survive 5 years? What share?
That's 5%, and like
5%.
if those you'd go look back to see what their pathology was to make sure it was a GBM. So. We're looking at recurrent GBM. The median survival in clinical trials probably 6 to 9 months, and newly diagnosed probably a year or two, depending on the other status. So there's not this kind of problem of waiting for the trial to mature and those kind of things. But that is a problem for other diseases, and we've thought about this. Statisticians have thought about this, and one analogy that Don and Scott Berry like to make is to baseball over time, baseball, hockey, and golf. Scott Berry wrote a paper comparing players from different eras over time through statistical modeling. And you can kind of use the same approach for controls over time. So you can't compare Mark McGuire to Ted Williams, but you can kind of like see what era they played in and what the kind of standards were and like those kind of things. And they do the same thing, and we do the same thing in GBM Agile, and they do the same thing in iSpy.
I'm surprised in Boston you're willing to compare Ted Williams to anybody, but
I'm a Detroit sports fan. So.
Right. In immunology, or in the approach that we've had in immuno-oncology, there is a feeling of N of 1 to some of these patients. I mean, you're taking, in CAR T, for example, it's not, we're taking individuals' own T cells out and then ramping them up and putting them back in and souping them up. How do you do that in a clinical trial setting? Isn't that really an individual patient's journey?
Well, CAR T cells, yeah. I mean, I don't even think we begin to know really how to conduct larger scale clinical trials with CAR T cells, right? I think for each individual patient is quite unique. And
so you're focusing on checkpoint inhibitors and what are the approaches?
So the immunotherapy field is broad, of course, and at the moment the focus is on checkpoint inhibitor combinations in early stage. And the challenge there is huge as well. I mean, there are ongoing 1,100 trials with combinations, but I think there's a need to really focus on rational combinations and putting those into master protocols that allow enrollment of small numbers of patients and really learning from what we're seeing from the correlatives and looking at outcomes and then feeding that back into a loop of how do we treat patients.
One question for all of you is this idea of the clinical trial in the platforms setting has changed because there really isn't a traditional sponsor, right? A drug company sponsor. And this gets into this issue of trusted third parties. We see so many of these organizations like CRI. Brian, you're starting one from scratch, right? To deal with how do you fund this kind of an organization.
Our experience on both Insight and GBM Agile, Insight is a phase 2 randomized screening trial, so there was less kind of problem with anything really. And Agile, as when we were progressing to be a registration trial, we worked very closely with FDA, and I think it helped that we didn't have any drugs signed up or anything like that. We were a nonprofit organization. We went to FDA. Scott Berry, Don Berry, and I sat down with FDA statisticians for 4 hours going over our models and like simulations. This is how we intend to estimate type 1 error. This is how we intend to graduate. We made some tweaks based on what they did. And we did that all. These trials take a lot longer to build. So like you need to have them ongoing to really realize the gain over time. And I don't think any one pharmaceutical organization that's developing one drug would spend that time. But we wanted to do it. We could do it. There was a little bit more exchange, I think, with FDA. And then now that we've gone out to pharma to get the drugs, we had a meeting about a year ago with these kind of adaptive trials where it shares information across arms, and Janet Woodcock was there, and I said, would you be able to approve the master protocol without any drugs? And she said, no, but we'd be willing to help in any other way that's possible. And I went back to her and I said, can you write us a letter or something so that when we're going to these companies, we can show some evidence that we're not just talking, because this is a stage 2-3 development in a pharmaceutical company, they're not going to just say like, okay, cool, like you said it. So like Janet gave us a letter. She had a letter addressed to our organization, said we've reviewed kind of your statistical design. There's no additional regulatory risk from your design, and that's been enormously helpful when we talk to companies, because they look at that and say, okay, we're not taking on more risk than we need to.
To whom it may concern, please excuse Brian for ... Right.
Yeah.
Okay. I would love to get some questions in here, comments. Bill, I know did you satisfy your question urge before?
I think that's
Okay.
I do think it.
We got a mic coming for you in a second. Yeah, thanks, sorry.
I think it segues from the previous conversation about using data to pattern analysis again for event prediction, but this is a step, it sounds like a step beyond, in that in trial designs, if we could find cohorts very similar to each patient that is enrolled in a phase 2 trial, and you could actually follow in real time if you're similar to the COMPASS trial, following these patients throughout their lifetime, and every time a patient went on the Agile trial, you found 10 patients in a database that looked like phenotypically, genotypically very similar, and follow them concurrently. And that would be an in silico, if you will, randomized clinical trial, but there's no randomization. We're just identifying and following. I just want thoughts.
It all comes down to signal and noise. If you have an endpoint with a lot of noise, then you're either going to need a really big signal or some good controls that are randomized. If you have one that's less noise, then you need less signal and you can look.
That's great.
And I think they're converging in the middle. The other thing, to your point, is if we could get trials to use the medical record. I mean, GBM, we care about what arm you are randomized to and how long you live. Like, how hard is that to get out of a medical record? And if you could just get that out of the medical record, the price of drug development would go tenfold less, because the monitoring and all the follow-up for all these kind of things, that's what costs $100 million and not $1 million.
The data were large enough and comprehensive enough. You could have your precision in matching a patient and developing a core could be far greater than a randomized one.
Yeah, so we've modeled that. We can talk offline, but we've modeled the kind of the actual variability and endpoints over time for different diseases and what signal you need and where you fall on those kinds of things.
I was going to follow up on that, Brian, because I think our senators, our House of Representatives, I think our patients think we already get that out of our medical records. It's an embarrassment for this country that we are wasting those data, because the idea that we need a trial in GBM where everybody's dead in 5 years. Rare patients survive longer than that. So if you come up with something that those patients get and 30% of them are alive at 5 years, you've got your registration. The FDA, this FDA in particular, would basically say that. So high quality registry data from the real world is something that I'm surprised the American public hasn't just demanded. No other industry would. If we said we don't know how many planes went down last year, that would be ridiculous. The FAA would be out of a job. So that's one point. The other point I would love you guys to just quickly discuss, or maybe for the Accelerator to address, is the idea that everybody's forming their own trial organization. Why do you need a trial organization? It's as if I want to make a widget. I'm going to build a factory. Okay, I made my widget. I'm going to tear the factory down. Now I want to make another widget. I have to build the factory up. Or in this group, I want to play a football game. Let's build a stadium, and then when the game's over, we'll tear the stadium down, and the next week we'll build another stadium. That just seems like a waste. So I'd love you to think about, why is that? We invest a lot in clinical trials, but it's somehow not a good return on that investment. That's the secret that nobody's talking about in the room as well, from a public policy standpoint.
It's interesting too, George, when I first met George in 2003, I went in your office and you were studying how a bizarre mutant Frankenstein fusion protein called BCR-ABL was influencing not one cancer, chronic myeloid leukemia, but gastrointestinal stromal tumor, two totally, completely different tumors that you would never see together in the same paper. And yet, so this raises the question of basket trials, and and what Lou was saying before about these individual, mechanisms, genetic pathways that may be driving lots of different kinds of diseases, maybe even two non-cancers or two different diseases. We've got 20 seconds left, 30 seconds. I do want to talk about this business model challenge for you, because as you build up a new trial infrastructure, how do you do that? I mean, where do you even begin to raise the money for that?
So raise the money through standard philanthropic kind of things for the initial raise. But then, to be honest, the business model, these trials are so expensive, is either a pharma fee for service, paying to like go put the arm on the trial, and like kind of the price that they would pay versus what they normally would is about a third of the cost, and it still would make us sustainable. The other thing we need to think about different models, and I've talked to Kathy about this before, is so we've talked to a company that has up through phase 1. They have a phase 2 dose. They're ready to go in. They're ready to go into our trial. They have no money. So how do we help them raise the money, or help them get in licensed into a pharmaceutical company? If we raise the money for them, can we get licensing? Can we get something else to help sustain? But if we had some war chest right now, if we were in a steady state 10 years from now, and we were doing like 50% of the GBM research, and you had to come here because it didn't matter, it's so much more efficient, then we could go and actually go get drugs that we want or fund research that we want and those kind of things, rather than just kind of sitting around waiting for somebody to give it to us.
The idea of the platform trial is also importantly to reduce the failure rates and to increase the success rates of the drugs that do graduate. And I was reading Andrew Lowe, our keynote speaker, has a great stat where he says that, if you look at normal phase 2 success rates transitioning out to an approved drug, it's like 30% or something like that. Those are emerging from a platform trial, it goes up to 85%. So it's a huge thing. And with that, I want to thank Brian and Laura and Amy. Thank you.